Yes, exactly.
So, the transcriptome part of this, okay, you found some really interesting stuff. Genes doing weird things in different species, and so what does the transcriptome part of this add to this question? What does it do to allow you to go further with these questions and some of these discoveries you had already done?
So we wanted to know if we could have a better understanding and maybe identifying novel genes that may be involved in integument development in gymnosperms that we may be missing from our angiosperm approach. So we started sequencing the different tissues of these species so that we could compare them. So for instance, in ginkgo, we dissected out the integuments, the megametophyte, the leaves, the pollen cones, and we sequenced everything separate. And again, with Taxus, we did the same thing. We sequenced the aryl separate from the ovule and the leaves and the cones. In Ephedra, we did the exact same thing. The bracts separate from the ovules and the leaves and the pollen cones. So we could compare which genes are expressed in the different tissues and identify genes that are differentially expressed in the structure of interest for us, which in every case was at the end a different structure. These are all different species, and the structure forming the seed code is going to be something different. So in ginkgo, we were looking at the integument. In Taxus, we focused our study in the aryl, and in Ephedras, we looked at the bracts that I mentioned before. Some of them become fleshy, so we included one species with fleshy bracts, and we included a species with dry bracts, which is Ephedra california. So we performed this transcriptome analysis. We sequenced the transcriptomes in these different tissues, in these species. And it turns out that we found that the genes that are expressed in these different seed codes are, again, different. So even within gymnosperms, the overall genetic network and the differential expressed genes are different. There were not many similarities in the genes that were expressed between these species. And we were able to identify some genes that we had no idea could be involved in integument development. So for instance, in ginkgo, we identified this gene family. We identified many different transcription factors that were upregulated, so they are highly expressed in the integument, for which we had never heard about being involved in integument development. Some of these gene families are very, very well-known gene families. For instance, we got the Ipedala2 gene family again.
Oh no.
And yes, but we don't know what Ipedala2 is doing in the integument in gymnosperm. And this happens with many different gene families. We found many large gene families, like members of the MATS-Box transcription factors, which is also a very well-known gene family. And we identified this family known as Fantastic Four, which is a relatively new gene family. It has been recently described, but is known to be involved in shoot apical meristem development in Arabidopsis again. So in Arabidopsis, it hasn't been identified in seed development, in integument development, but we found the gene that is highly expressed from the transcriptomes, a homologous gene of a Fantastic Four. And we did the expression pattern, and it turns out that is specifically expressed and strongly expressed in the integument in ginkgo. We didn't find the expression of this gene anywhere else, not in the pollen cone, not in the leaf, only in the integument, which allowed us to not only corroborate our transcriptome data, but also to propose this gene as a possible gene involving integument development in gymnosperms, and to show